All right. Changes in chromosomes. So the first two that we talked about yesterday, one of them we said that if there was a total non-disjunction that you could end up with extra sets of chromosomes, and that would be called polyploidy. Okay? Polyploidy means that you have extra sets of chromosomes. Homologous chromosomes. Okay, so you could be 4N, oopsies, 4N, yeah.
Is that supposed to say homologous?
Homologous, thank you. 4N, 8N, 12N, something like that. Okay? If you don't have a full non-disjunction, if you only have non-disjunction in a single chromosome, you end up with something that's called aneuploidy. Okay, and aneuploidy is what we talked about yesterday, a trisomy, or the other cell could also be inherited, and that would give you what's called a monosomy, because you'd be missing one of the chromosomes there. Okay, so that is an abnormal number of one chromosome. We'll say chromosome type. Okay, and so we could have, in this case, a monosomy, which is only one chromosome of that type. Or you could have a trisomy, which would be 3 chromosomes of that type. Okay, and then here is a list of the aneuploidies that can happen in humans, right? So on the left side, you've got what they've listed as monosomies, but they're not all monosomies. Some of them are caused where part of the chromosome actually got deleted or removed. So you still might have 2 chromosomes, but one of them's all wonky. And then on the other side, you've got all the trisomies. And the trisomies are the trisomies of each individual chromosome. So there's a chromosome number over here, and here are the different trisomies. So for the most part, trisomy 9, the different colors on here, if it's red, that means that they never survive to term if they have a trisomy of that type, so that'll be a miscarriage. If it's yellow, that means that most of the time they don't survive to term. And if it's green, that means that they can survive to term, or they normally do, but they will rarely live after that point for more than 2 weeks or so in the case of trisomy 9. And then also in Down syndrome, that they live longer than that, but with a host of problems. Okay, so you can see here that the trisomy tends to be really bad. Okay, most of the time trisomy results in death. The one case where it doesn't result in death is Down syndrome, right? And so that's the one that we really are familiar with. Is Down syndrome, because it is a result of trisomy of the 21st chromosome, and so we say that Down syndrome is trisomy 21. Okay. The other thing that I want you to notice on here is that this only goes to 22, right? But you have 23 chromosomes. This is because the 23rd chromosome, as Derek was saying from Veritasium, that the 23rd chromosome is a sex chromosome. And so up here are all the differences in what you can see with the sex chromosome. So males are XY, females are XX, and so the X chromosome turns out to be really, really important. It's got like vital information on it. So the ones that are lethal over here, I know you can't read it very well because it's red on black, but these are all lethal because they're only Ys. They're only Y chromosomes. And if you have only Y chromosomes, then you don't have the X chromosome, and the X chromosome is needed to live, and so you die, right? But there are some other interesting things on here, right? So you can see this is a monosomy of the X chromosome. It's X0. So there's no Y chromosome, but there's no other X chromosome. And so based on what Derek was saying, that shouldn't really matter all that much, right? Because we know that the other X chromosome is just going to be inactivated anyways. And sure enough, we see that people who have this are generally fairly healthy. They have Turner syndrome, but that tends to cause problems when it comes to puberty. So people who have Turner syndrome present as females. They look like females, but they don't normally go through puberty. They just look prepubescent forever in that case. There are also some interesting situations where people might have an abnormality that they don't know about. Okay, so XX is the normal female, but XXX, that would be caused by non-disjunction in an egg cell, or possibly non-disjunction in anaphase 2 of a sperm cell, right? Presents as a normal female. People would not know that they had XXX syndrome, right? Unless they had a karyotype run on them where they actually looked and said, hey, you have extra chromosomes of your X chromosome, right? Because the bar body, that thing that is the inactivated X chromosome, will then just have 2 bar bodies instead of 1 bar body. And so you still have the same genetic makeup then as a normal female, but in case of what we're talking about with the stripy females, in case you weren't following along, right? There are parts of the body that have one X chromosome inactivated and parts that have the other X chromosome inactivated. But if you were XXX, you would actually have 3 different things, 3 different chromosomes that were being expressed. And that's only if both of the X chromosomes came from or if the non-disjunction happened in the egg, because then you could have 2 different X chromosomes. Whereas if it happened in the sperm cell, they would be identical. Yeah?
But like, going off that analogy, would that mean they have like 3 different color stripes?
Yeah, exactly. Okay. Yep. All right. Same thing goes down here with males. Normal males are XY, but you could also have XYY. And so XYY it says it presents as normal male. It's got a name. It's called Jacobs syndrome. And people with Jacobs syndrome generally don't know they have Jacobs syndrome. You look and act just like a male. The only thing that I've heard is some of the male characteristics, wide shoulders, deeper voice, can be slightly over-exaggerated in somebody with Jacobs because they have extra X chromosome or extra Y chromosome stuff being expressed. Yeah.
So there's really no problem with that?
Nope.
Like you wouldn't know ever?
Nope. Okay.
So according to non-disjunction, the difference between poly and
Aneuploidy?
is that one polyploidy occurs when non-disjunction occurs in 2.
Okay, so imagine that you were talking about a human cell. Okay, so a human cell has 46 chromosomes. Normal human sperm cell will have 23 chromosomes then. Okay, if it has what's called a complete non-disjunction, that means none of the chromosomes separated. And that means that the sperm cell, instead of having 23 chromosomes, will have 46 chromosomes. Okay, that would cause a polyploidy if that thing was able to then reproduce with another 46 chromosome individual, okay, or 46 chromosome egg. But in the case of aneuploidy, it's a single non-disjunction. So if a normal human cell has 46 and a normal human sperm cell has 23 chromosomes, then one that was caused by a single non-disjunction would have either 22 chromosomes or 24 chromosomes. If it had 22 chromosomes, you'd be looking at a monosomy in the offspring. If it had 24 chromosomes, you'd be looking at a trisomy in the offspring. Right? Okay. Then there's some other things in here, Klinefelter syndrome. So these are the ones that are mostly survivable. Trisomies of the sex chromosome are relatively non-lethal, whereas trisomies of non-sex chromosomes tend to be very, very lethal. Okay, so we can say that sex chromosome trisomies are far less lethal and then or I guess trisomy and monosomy. But yeah, you get the idea, right? Okay, so this is if the whole chromosome is there or the whole chromosome is not there. Okay, you can also have situations where part of it's there. Okay, so you could have a deletion. Okay, you can probably figure out what a deletion is. That's when part of the chromosome actually gets removed. Okay, so if we think about a chromosome like this, and let's just say, for the purpose of discussion, that this chromosome has segments A, B, C, and D on them, and let's say each of these segments has, I don't know, 500 genes or something like that on it. All right. A deletion would be if you, let's say, got rid of one of the parts, and then you were just left with a chromosome that had A, C, D on it. Okay. Where did B go? B got removed. Is that going to be a problem? Probably. B was probably had some important information on them. Is this person going to die? Possibly. Why wouldn't they die? Yeah.
Because they have duplicated chromosomes, so there's an extra set.
Yeah, because there's a non-deleted chromosome also, because you've got two sets of your chromosomes, right? And so even though this one got deleted, so it's missing part of it, then the other part is still making up for that lost work. Okay, this is sort of how genetic disorders work. This person that had a deleted area here could be fine. You could never know that they had a problem, because if their one chromosome is able to produce enough of the stuff that they need for that, you know, whatever that enzyme did or whatever that protein did, then you'd never know that they had a problem. Okay, but if they then reproduce with somebody else who has the same mutation, and that's the chromosome, the deleted one gets passed on in one person and the deleted one gets passed on in the other person, now they have no copies, no working copies of that gene, and that person's probably going to die, right? Or that person's going to have a serious genetic disorder, right? That's where the whole inbreeding thing comes from. The whole thing about how if you inbreed, you have more genetic disorders. It's not because it produces any more mutations than anything else does. It's that you're more likely to have the same mutation as the person that you're reproducing with, because you are coming from the same genetic line, right? And so, you know, let's say I had a random mutation where I lost segment B, and it just happened last generation, like in my mom's DNA or something like that, right? I might be the only person on the planet that has that genetic disorder, right? Except for my sisters. They might have it too, right? So as long as I don't reproduce with my sisters, my offspring is never going to get that genetic disorder, because they're always going to have one working copy, right? But then my offspring then might have it, and then their offspring. And so the more removed you are from your offspring, from your previous offspring, the less likely you are to have the same genetic disorder, right? All right. Duplication. That is when multiple copies of the gene are on the same chromosome. So multiple copies of the same gene on a single chromosome. Okay, so let's say that we have this chromosome here, and it's A, B, C, and D, and then it gets duplicated, and now you have actually, let's see what it's A, B, C, D, or something like that, right? It could be A, B, C, D, right? And what's the result of that? What does that do?
Would it overexpress?
Yeah, you overexpress. You produce a bunch of whatever that protein is. Okay, and so if a normal person has 2 copies of it, let's say they produce, 200 units of it, right? But if a person has 10 copies of it, then they produce 1,000 units of it, and that's probably not good because evolution has left us with the correct amount of most of our enzymes and proteins. Okay. Translocation. One portion of a chromosome is moved to a non-homologous chromosome. Okay, so let's say we have A, B, C, D chromosome, and then we also have thin chromosome that has W, X, Y, Z genes. Okay, and then there's a weird translocation here where you end up with A, B, C, D, and then W, X, and then Y, Z are over here. What's the result here? This one is a little bit more difficult to think about. Yeah. What do you think?
What's the difference between why is one like skinnier than the
Because they're non-homologous chromosomes. It would be like, let's say, chromosome 1 and chromosome 2 in your body, right? And part of chromosome 1 ended up on chromosome 2. So the immediate result of this is the tricky part. The immediate result could be nothing, right? Or what if this is a chromosome that's inactivated in certain cells and active in other parts, right? That means that if the whole chromosome is going to be condensed into really tight chromatin so it doesn't get expressed, but now it has important genes on it, those cells might lack an enzyme that they need. Or if you think about it the other way, in parts where this is supposed to be inactivated, right, but it is activated, it's now producing 2 extra proteins in there that shouldn't be made in those types of cells. And so the cells themselves are going to be different than what you would expect them to be. Like an epithelial cell might also be producing digestive enzymes or something like that, right? And that's probably bad. That's the immediate result, if there is any. What's the more long-term result? Yeah.
Maybe like a deficiency in some area.
Again, that's more immediate.
If you pass it on to like your offspring, it wouldn't be necessarily like a new species, but like it would be like a huge mutation.
It would be a very big mutation, right? So let's say that we do pass this on. Let's say we pass this one on. That means that they're going to have extra WX chromosomes. They're going to be essentially trisomies, not WX chromosomes, WX genes. They're going to have essentially a trisomy for these proteins that are being produced here. Okay? What if you pass this one on? Then they're going to be deficient in those. They're only going to have 1 copy of those. Right? So this is going to really change things. Okay? This is a sort of partial of what can happen. If it happens with the whole thing, we call that a fusion. Fusion is when 2 chromosomes, 2 non-homologous chromosomes, join together. So like this. This is a big deal. Okay? This is the biggest difference between us and great apes. Is that we've got this fusion, right? Fusion of chromosomes. Same genes, just chromosomes fused together. Okay?
By non-homologous, like, is it referring to the paternal and maternal?
No, non-homologous means like chromosome 1 and chromosome 2.
Oh, okay.
Yeah.
Makes sense.
All right. And then the last thing would be an inversion.
Wait, so you have 23, so it'd be like chromosome 1 fusing with chromosome 2.
Yep.
Would it be like 1 and then 20?
Yep. So how would you spot that? How would you, if you were a researcher, how would you know that a fusion had happened? Yeah.
The length of the chromosome would change.
Length of the chromosome changes. That's one thing. Yeah.
The shape of it, like some of them are thin.
Yeah. So the shape of the chromosome changes, but even more related to that, the thing that changes the shape is the centromere, right? So where are they hooked together? How many centromeres would you see on this thing? 2 centromeres, right? Also, we know, maybe you guys remember this, when we talked about cancer, it talked a little bit about the telomeres, the segments on the end of the chromosomes. So this has a telomere, this has a telomere, this has a telomere, this has a telomere. That means you're going to find a telomere here, and a telomere here in the center of the chromosome, right? And that's what we see. 2 centromeres, telomeres in the middle, yeah.
Would the telomeres in the middle be expressed?
Nope. You have a lot of non-expressed DNA, so that's not weird to have non-expressed portions, but it's just, it is identical to the telomeres. All right, inversions. That's when genes switch places on a chromosome. A, B, C, D becomes A, C, B, D. What's the immediate effect of this? Probably nothing. What's the long-term effect of this? When would this ever come into play? Yeah.
During crossover?
Only during crossing over, okay?
So,
If you cross over here and it exchanges and you end up with one that's A, B, B, D and one that's A, C, C, D, right? Then you now have both a duplication and a deletion, right? Questions on that?
wait, why would you have a deletion as well?
Because the B wouldn't be there anymore. You'd have 2 C's but no B in one of them, and then you'd have 2 B's but no C in the other one.